Good afternoon, everyone. It's really my privilege to be one of the cancer researcher to be here and present some of my work. Firstly, I would like to introduce my research journey. I received PhD from Monash University and have undertaken a couple of years of postdoc training in ANU, and I moved to UQ. And recently, I was honored to be recognized by UQ Hero Program to establish a new research program team, which centralizes on the understanding of T-cell immunity and apply the knowledge to improve vaccine design and engineer next generation of immunotherapies. Why is T-cell immunity so important? A major function of T-cells is to directly kill the pathogens and tumors. This is mediated by a specific subset of T-cells called killer or cytotoxic T-cells. So they express a signature protein called CD8 on their cell surface and usually called CD8 T-cells. So you can see from here, they can recognize the cancer cells from the healthy ones and then destroy them. Indeed, in most cancers, the abundance of tumor-infiltrated CD8 T-cells is positively associated with the prognosis. However, CD8 T-cells in cancers are very prone to a dysfunctional state called exhaustion, and as they have been persistently exposed to overwhelming amounts of tumor antigens. Besides, tumor also express some special ligands that can activate inhibitory signals in T-cells. The exhausted T-cells will gradually lose the function and cannot effectively control tumor growth anymore. In the past decades, a major breakthrough in cancer therapy is that scientists found if we can block the inhibitory signaling in T-cells, such as PD-1, then we can effectively revitalize T-cell function from exhaustion. So this strategy is called immune checkpoint blockade therapies, and they have successfully prolonged the survival rates in many cancer patients. However, as we saw from many projects presented in this afternoon already, in most cancer types, the efficacy of immune checkpoint blockade therapies remain to be improved. This issue is highlighted by what we observe in lung cancer, which is the most common cause of cancer death in Australia. In lung adenocarcinoma, which is the major form of this disease, the overall response rate to the immune checkpoint blockade therapy is below 20%. The major question we want to address in this cancer project, therefore, is to identify a new approach to boost T-cell immunity and improving the cancer therapies. To address this question, we need a more comprehensive understanding of anti-tumor immunity beyond CD8 T-cells. In the tumor immune cells landscape, there are many more other cells that can impact CD8 T-cell functionalities. In particular, these CD4 expressing T helper cells, which can literally provide essential helper signals to CD8 T-cells, are also very important in affecting cancer patients' overall survival and their response to immune checkpoint blockade therapies. In particular, these specialized subsets of T helper cells called T follicular helper cells, TFH cells, have been demonstrated to be required for the anti-tumor immunity in lung adenocarcinoma. In this recently published published paper in Cell, the authors identified that the increased TFH cells infiltration in the tumor is positively associated with the better survival in the patients. But the role of TFH cells in anti-tumor immunity wasn't very extensively studied before because this population was more commonly associated with the B cells function in the antibody response. But more and more evidence suggests TFH functions are very important for regulating anti-tumor immunity. Regarding the biology of T follicular helper cells, my previous work has led to several impactful discoveries, which includes two co-first author papers in Nature Communication and Nature Immunology, which was published as a cover story. So here we hope to leverage such expertise into our understanding of the T-cell mediated anti-tumor immunity. So how does T effector cells can enhance the anti-tumor response? One of the major mechanisms is to produce important cytokines, which is highlighted by the role of interleukin-21, IL-21. So IL-21 is very unique compared to among various cytokines. Two landmark studies in 10 years ago have shown that IL-21 can selectively suppress T cell exhaustion in chronic viral infections. And then a later study demonstrated that the major source of IL-21 in this scenario is T follicular helper cells. And importantly, as the cell paper I mentioned, they demonstrated that the deficiency of IL-21 signaling in the mouse lung cancer model will severely dampen T cell anti-tumor efficacy. So given IL-21's promising clinical benefits, IL-21 alone or in combination with other drugs have been tested in various clinical trials. However, the overall response rate is not satisfactory, which at least is partially due to the cytokine's low in vivo efficacy and the dose-limiting cytotoxicity. So can we find a way to improve IL-21's efficacy to really overcome such limitation in a therapeutic application? That's a big question we ask. So in my recent work, we identified that the bioactivity of interleukin-21 can be regulated by a conserved cationic residue cluster, meaning these amino acids, they have high density of positive charge. And if we mutate them with into a neutral amino acid, then the function of IL-21 can be drastically increased. This result indicates that the CFC region can be a promising target to modulate the function of IL-21. So by harnessing these unique biochemical feature of IL-21, we develop a novel antibody which can target the CFC region and significantly increase the function of IL-21 by over 10-fold. We also conducted mechanistic studies to review the unique structural changes in interleukin-21 by binding to this agonistic antibody. We further tested the in vivo efficacy of this agonistic antibody in the mouse tumor model of MC38 colon cancer. The tumor transplanted mice were treated with PBS, IL-21 alone, or IL-21 in complex with the agonistic antibody for multiple days. And here we can see IL-21 alone actually failed to induce, clear anti-tumor effects. However, when it is in complex with the agonistic antibody, there's a very unique anti-tumor effect, where the majority of tumors were suppressed or eradicated by day 15. We then observed that IL-21 in complex with agonistic antibody can selectively enhance T cell functionalities as measured by their capacity to producing important cytokines such as interferon gamma. And then we also observed that IL-21, when it's in complex with the agonistic antibody, can effectively suppress exhaustion status in the T cells. Last but not least, we recently also tested the synergistic effect of our agonistic antibody with the immune checkpoint blockade therapies in our mouse tumor model. And we can say it really can has the promise to boost T cell functionalities in addition to the PD-L1 blockade therapies. So supported by the critical role of IL-21 in the anti-tumor response of lung adenocarcinoma and the promising efficacy of our agonistic antibody, we hypothesize that this novel antibody can improve the anti-tumor efficacy of immune checkpoint blockade therapies in lung cancer model. So in the following 2 years, I will firstly evaluate antibodies' therapeutic efficacy in a lung cancer model, focusing on how it can enhance the efficacy of immune checkpoint blockade therapies. We will then generate a novel bispecific therapeutics that combines agonistic antibody with immune checkpoint blockade therapies. At the end, I would like to acknowledge my supervisor and the help of the team member. I would also like to express my gratitude for the support of the Q Cancer Foundation and all the amazing funding bodies again to enable this project to happen. I'm also like to especially thank the consumer advocates for the strong support and the empowering advices. Thank you very much for your attention.